about the past 6 years, which is at the interface of engineering and biology. So my background is in engineering. I'm an electrical engineer, and my background's in control theory. So I work in, well, first of all, building mathematical models of systems, and then using these models to help understand how to manipulate systems to get them to do what we want to them to do sort of better than they're doing now. So we do a lot of work with aircraft, with NASA on air traffic control systems, so a lot of engineered systems. And about 6 years ago, I guess now, I got very interested in applying these tools to not really in the design part, but more in the understanding of biological systems. And I've been particularly interested in protein regulatory networks, and I'll kind of describe what those are. This is joint work, so it's a collaborative effort between me, my group, and that of Jeff Axelrod. He's a developmental biologist. He's a professor in the Stanford University School of Medicine, and he works in different areas related to development, but his main lab is centered around flies, Drosophila. He has an experimental lab where they study the development of flies.
Are you going to show photographs and make us throw up our hands?
Yeah. Well, they're not really. I mean, I think they're really nice pictures, but. And then and a bunch of really great students. So Keith and Robin and Dolly have just finished their PhDs at Stanford. 2 of them are in engineering. Dolly's in the cancer biology program. And Anil is a PhD student here at Berkeley. Okay, so this is what I call a very stable and robust system. And this is, I think it sort of pictures the Holy Grail of developmental biology, trying to understand how human embryos develop. And all of the intricacies of that, so the details of how cells divide, how cells differentiate. So how do cells that are all initially the same, these stem cells, figure out which cells are going to become skin, which are going to become hair, which are going to become fingers, etc. And not only just the developmental process, well, in understanding the developmental process, it's sort of hoped that one could understand how diseases develop. And if we understand how diseases develop, then potentially we could use that knowledge to design interventions or to actually use control to help mitigate these diseases at the genetic level. So this is, I think this is the Holy Grail, but this is a very, very hard problem. It's a huge, complex problem. And so biologists, if you go to developmental biology and you sort of look at different professors in their labs, you'll see that biologists focus on smaller systems, systems that they can study and systems that they can do experiments on. And so I'm going to talk about 2 systems today that I've worked on. One is the fly, and that's the work that we did with Jeff. But the first is, so I'll talk about the fly first, and then I'm also going to talk a little bit about another system we've worked on, which is a frog embryo. And you sort of see this if you go to biology departments, that there's a lot of work being done on flies, on worms, on zebrafish, on frog embryos, because these are and mice, etc. These are systems for which there's a fairly short life cycle. So when you form a genetic mutant, you don't have to wait too long before the adult organism develops, and so you can sort of see the effect of what you've done. Okay, so I'm going to start by talking about the main system we've been working on, and that's flies. So this is a fruit fly, a Drosophila melanogaster. There's an image of it. You've all seen them. These are the little fruit flies that you see, but there's an image there on the upper right-hand corner. And the particular part of the fly that we've been studying, and this is the majority of what Jeff's lab has been doing, is studying the development of the fly wing. So that's pictured up in the upper left-hand corner. It's just the wing laid out. And the proximal side. So proximal means it's the side of the image or the side of the cell closest to the fly body. and that's on the left-hand side. Distal is the side of the image, the side of the cells, et cetera, closest to the wing tip, and that's on the right-hand side. And then just to note that all of the images, when we talk about flies here and fly wings and cells in the fly wings, all of the images in my talk are going to be oriented so that the proximal side is on the left-hand side of the image and the distal side is on the right-hand side of the image. Okay, so in the lower left-hand corner, I've blown up a little part of the wing. And what happens in the development of the wing as the fly is in its puparium and it's developing? So the wing cells are dividing, and there's at a certain stage of development. It's about 15 to 18 hours after the puparium forms. The wing is sort of formed. All of its cells are there. And then this process by which the wing turns into an adult wing, an adult fly wing, starts. And that process goes between 18 hours after puparium formation to about 30, 35 hours after puparium formation. And around 35 hours after the puparium forms is when the adult fly, well, it's the embryo hatches, the puparium opens, and the fly is finished. So during that time period, a very interesting process happens. And that process causes a little hair to grow from every cell on the wing. And you can see that. Those are the little black lines on the image. And I've got a sort of stylized view of some cells in a sort of patch of cells. In each cell, the hair always grows on the distal side of the cell, and it always points distally. So, and so we can think of this wing as two planar arrays of cells. And so you can sort of see that. They're like these prisms, and you see there's actually a repeated patch of cells on the underside of the wing. And so the point here is to try to understand how the cell, or the sort of interesting problem here for flies is trying to understand how a single cell in these tens of thousands of cells, so a single cell understands which is its distal side, so it knows where the hair is supposed to grow. This is a very robust system, and under all sorts of conditions, under all sorts of temperatures, the flies develop this way. And it's an interesting problem for flies. I mean, it's important for the flies to have a smooth array of hairs over their body because it improves the hydrophobic property of the fly. Little raindrops will just roll off the fly wings and fly body if there's a smooth array of hairs. But that's really not why people study this mechanism. The problem of how cells determine which side is which, or in this problem that's termed planar cell polarity, how cells determine their polarity, is very important in development in general. And it's not just important in flies, like this example that we're looking at here, but it's sort of a ubiquitous mechanism that goes on in all embryonic development, in human development. So in cell differentiation, it turns out in many different types of cell differentiation, it's very important for cells to have a sense of their polarity so they know how to divide. And just a couple of examples I've got on the bottom right-hand corner of human pathology. So these are diseases in humans that are caused by a disruption at the genetic level of the cell polarity circuit. So there's a disease called Usher syndrome, which is it's in human embryos. The hairs in the inner ear, in the cochlea, are malformed, and the baby is born deaf. So it's a congenital deafness syndrome, and it's related to cell polarity. In fact, some of the same proteins that we study in flies, there are human forms of those proteins, which has been shown that if those proteins are lacking in the ear development, then the ear won't develop properly. This disease called spina bifida, it's again a disease in human embryos in which the spinal tube doesn't close properly and the baby is born malformed. That's a problem. So the failure of the spinal tube to close is a problem of cell polarity. And there's a relationship to a cancer pathway, what's called a canonical cancer pathway involving one of the same proteins called Wnt, which we study in the fly. So there's a relationship to some cancer pathways as well. So the idea is trying to understand this particular circuit will help us understand the development of flies, but the real goal then is to use that in turn to help understand the development in humans. And again, we're looking at a very particular circuit, but it's a very important circuit. Okay, so I'm going to show you some data, and this is data from Jeff, my collaborator, Jeff Axelrod, his lab, as well as other people in the community. There's a fairly large community of people in developmental biology studying cell polarity, and it's spread around the world. So these are some images of, they're actually of cells and a fly wing. And the cells they're basically like bags of fluid. So when you pack them in a plane, the most efficient way to pack them is as sort of hexagonally packed. So in a regular cell array, each cell is touching 6 other cells. It's 6 neighbors. So that's how you sort of see these hexagonal patterns for cells that are emerging. So there have been a number of experiments done over the past really 15 or 20 years studying cell polarity. And the kinds of experiments that biologists do are they knock out the function. They hypothesize that a certain protein is implicated in the mechanism, in cell polarity. They knock out its function, and then they observe the effect. So then the fly develops. So they're able to, using heat shock or other mechanisms, to knock out the function of that protein in certain cells. And then all of the cells that divide from that cell don't have that protein function. And then the fly, hopefully, will just grow and develop normally. Hopefully, this doesn't cause the fly to die. And then they study the effect of that genetic mutant on the adult fly wing. And so what the first kinds of experiments that I'm going to show are where so they sort of understood a few of these proteins are important in cell polarity. And then they sort of looked at what happens to these proteins in the normal developing wing. So I'll show you the mutant cases afterwards. And in doing these experiments, they identified that there are 4 key proteins that seem to be very important in cell polarity. And I've got pictures of all 4 of those on the bottom. Their names are disheveled, frizzled, prickle, and Van Gogh. So they all have kind of funky names. There's a very interesting nomenclature system in biology. So the lab that discovers a protein gets to name it. And sometimes they name it after the lab, but usually they name it after the mutant phenotype for that protein. So Van Gogh protein, for example, if you knock it out, so you form a cell that doesn't have Van Gogh function, it's basically the function of Van Gogh protein is messed up, then Van Gogh tends to appear in these little star-like structures. So you get these bright spots around the cell. And so Van Gogh has another name, which is called Starry Night. That's its other name. So it's sort of an interesting nomenclature. Anyway, so we've got these 4 proteins. And in the normal developing wing, what happens? So disheveled and frizzled. So the bright spots here are they're a fluorescent protein tag that's attached to the particular protein that we want to study. So disheveled and frizzled localize to the distal side of each cell. And these images were taken just before the hair starts to grow. So about 30 hours into that window that we were talking about earlier. And prickle and Van Gogh, 2 of the other proteins, localize to the proximal side of every cell. And after a number of other experiments, it was sort of determined that the hair starts to grow wherever disheveled is localized highest. And it's interesting. It's like this sort of modular development system where there's this external timer that's going on. And so the circuit that's making these cells localize to different parts of the cell, it goes on from, as we talked about, 18 to 30 hours. And at 30 or 32 hours, the timer stops and says, okay, wherever disheveled is now, we're going to grow the hair at that point. So it's not like the system is waiting for disheveled to come to the distal side of the cell. It's like this timed process where one module stops and then the next module starts. And this is interesting because these proteins were all, if you look at them at the early stages of development, they're all homogeneously distributed throughout the cell or the cell membrane. So something's happening which causes them to localize asymmetrically. And so then the question went back to how does, of planar cell polarity, went back to what is the mechanism which causes these proteins to localize asymmetrically throughout the cell? Okay, so that's one piece of evidence. And then the second piece of evidence are these mutants that I was telling you about. And these are, the experiments that kind of, as an engineer or as a control person, they kind of make you excited about studying this. It's very, very interesting. So inside, this is a sort of a patch of cells on the wing, and here we're not showing the cell boundaries, but we're showing the little hairs. Inside the black line are cells that don't have the function of the protein frizzled. So there was a heat shock experiment done on one of these cells, and then all cells that divided from it are lacking the function of this protein. And so we see, if we look a little carefully, that the cells inside what's called this clone, mutant clone, those cells, they're kind of screwed up. They have 3 hairs. If you look at the cell boundaries, those hairs are growing from the center of each cell. And that's, we'd expect that because they don't have one of the key signaling proteins that's necessary. But what's interesting is that the healthy cells, the ones that have all of the requisite proteins, and on the distal side of the clone, those cells have reverse polarity. So the mutant cells here have somehow affected the neighboring healthy cells. And it only appears that the disruption is on the distal side of the clone. If you remove Van Gogh protein, you see the opposite effect, that polarity is disrupted on the proximal side of the clone. So as an engineer, and as an engineer interested in local control systems and sort of interacting control systems, one looks at this and thinks that there must be some kind of message passing or information passing from cell to cell. And that's the first thing that we as engineers might think, or as people studying sort of distributed control systems, for example. But the first thought that a number of biologists looking at this, they thought, well, the disruption of these cells must be caused by some kind of diffusible protein that's diffusing from the unhealthy cells to the healthy cells. And likewise, here there must be some diffusible protein that's diffusing onto the proximal side of the clone. And the problem was that no matter how hard these biologists looked for these proteins, they couldn't find such a diffusible protein. And so they named these proteins factors, like factor X and factor Z, just because they couldn't find them. So that's sort of some evidence. And so when I started working with Jeff, he had just proposed a different model. A model in which you don't actually need to invoke a diffusible factor. It's just information passing from cell to cell. And so the model is up in the upper right-hand corner of the slide. And the model is, this is a typical biological model. So it's basically an influence diagram which records positive and negative influences between proteins. So an arrow here indicates that there's a promotion effect. One protein will promote the production of another. And the other symbol up there, which is the arrow with the little line on top, up here, for example, that indicates an inhibitory effect. So in that case, what we're saying is the protein prickle is inhibiting the recruitment of disheveled protein to the boundary of a cell. So I've got 2 cells depicted there, but you should think about this interaction going on between all cells in the network and between each cell and all of its neighbors. So let me just say in words what this model is saying, and I'll use the cursor to illustrate that. So what the model is saying is that, So the protein frizzled is promoting the recruitment of disheveled protein to a boundary. So disheveled here is a cytoplasmic protein. Here I'm using the little baubles to represent a protein that only exists in the cytoplasm of the cell, whereas frizzled and Van Gogh are both membrane proteins. They actually wind in and out of the membrane. So frizzled promotes the recruitment of disheveled to the boundary. Disheveled further stabilizes the frizzled localization to a part of the membrane. And through a potentially different mechanism, frizzled promotes the recruitment of Van Gogh and prickle to the membrane of a neighboring cell. And prickle and perhaps Van Gogh inhibit the recruitment of disheveled to the boundary. And then you see the symmetric interaction on the other side. There's another piece of information contained in this model, which is the size of the font. So the large font indicates where these proteins tend to localize in the normal wing, what we call the wild type wing. So the disheveled and frizzled, as we saw, tend to localize to the distal side of the cell, Van Gogh and prickle to the proximal side of the cell. And so Jeff proposed that, and this was just about when I started working with him, that that model is sufficient to explain all of the data that had been observed about this particular system, all of the wild type, all of the mutant data. the other point is that this is, if we look at it carefully, it's a local feedback loop. It's something that we see all the time in control. And so to break the asymmetry, there must be some kind of directional cue, maybe a very, very weak signal indicating what is distal and what is proximal. And after doing a number of experiments, and this was actually done in the course of our studying this system, a fifth protein called FAT was identified to probably be this global directional cue. So there was a series of experiments that Dali Ma, she's one of the students that I mentioned, had done, where she did the same kind of mutant clone experiment. She knocked out FAT from groups of cells, so the cells inside this yellow line, and basically observed whether or not the polarity was disrupted. And what she saw were some very interesting results. If you look at the upper diagram, the red here indicates the hair, so it's the actin bundle that forms the hair. The lack of fat in the upper diagram doesn't seem to have disrupted the polarity. Everything seems fine. The cells know which is the distal, which is the proximal, and which is the distal side. But in the larger clone, this one below, the global polarity has been disrupted. You see these sort of characteristic spiral patterns. And this indicates to us that cells are maintaining their local polarity with their neighbors. So they're trying to align their polarity with their neighbors, yet the overall sense of what's proximal and what's distal has been lost. So in trying to understand the system, we not only wanted to really understand the original circuit that Jeff had proposed, but also any model that we propose has to account for the variability, like why in some cases is polarity maintained and in some cases it isn't. Okay, so here's just an arrow indicating that FAT is the protein we think that's providing the global directional cue. Okay, so that was the overall model. And then Jeff published kind of this influence diagram several years ago. And this is this community, I think it's sort of this planar cell polarity community within developmental biology is kind of contentious. And, I thought being in engineering was sometimes, competitive and, there's sort of friendly competition that goes on, but it's nothing compared to this developmental biology community. A very nice community, but, the papers they publish against each other. So as soon as Jeff had published this paper, there were all of these other papers that were published in sort of, like same-tier journals, which said, oh, there's no way this model could be correct, right? Because the first person to discover how it works is, that's kind of what you wanted. You want to be the first person who discovers how, or the first group to discover how a system works. And so there were all these not competing, but models that said there's no way, or papers that said there's no way this model can be correct because it's ignoring and it doesn't explain several other pieces of data that we've observed in the literature. So some sources of controversy, and I'll try to explain what these mean. If I showed you an image a few pages ago that if we knock out frizzled, we see this disruption of polarity. That's what's called a non-autonomous effect. Non-autonomous means that a cell's behavior is determined by something outside its own genes. So this disruption in polarity is termed domineering non-autonomy. But if you do the same experiment with disheveled, so you knock out disheveled, I didn't show that picture, you don't see any non-autonomous effect. There's no disruption of polarity around the clone. And people are looking at that diagram saying, well, disheveled and frizzled have a very similar role in that overall feedback loop. So why is there such a great difference if you knock out one versus the other? So that was the first source of controversy. The other is that there's some slight changes. If you take that frizzled protein and you form what's called a slightly different allele, so different version, some slight changes in the amino acid sequence, you form, and then you knock that protein out instead. So you've got a fly with this different frizzled allele, then you knock out that frizzled allele. You don't see this non-autonomous phenotype. You still see this, you see no disruption in polarity. So they wanted that explained as well. And actually the biggest source of controversy was the last one. And this is nicely described when we think about control. So a kind of senior level control theory class. If you overexpress, so you can do experiments where you not only knock out the function of a protein, but you can bump up the function of the protein by overexpressing that protein, forming cells that have more of that protein. So if you take a bunch of cells on the wing and you increase the concentration of prickle protein in those cells, then if you look locally at that feedback loop, it indicates that since prickle inhibits the recruitment of disheveled to the boundary, if you increase prickle, you should see less disheveled at the boundary. But the experiments actually show the reverse, that if you increase prickle, you see more disheveled at the boundary. And so that was really the biggest source of controversy. How does this model, if you just look at that sort of local interaction, that model seems counterintuitive. So this is where we started. And really, I think this is the main point that I wanted to get across today is the reason why we'd want to mathematically model biological systems and what you can get out of such a model. So, and this seems to be true in a lot of developmental biology systems. We're now working with a number of other systems where the hypothesis that the biologist has about how the system works exists at the molecular level, like I showed you in that previous model. Basically an interaction between molecules, between protein molecules. But the data usually exists at a higher level, at a tissue level. So there's a gap between the molecular level understanding and its tissue level effects. And so what a mathematical model, and that's where this intuitive gap, so somebody looking at that model has to sort of jump over that intuition and try to say, well, okay, I can look at that model and sort of liken that to the data. It's hard to do that. So a mathematical model actually can bridge the gap between the molecular level understanding and the tissue level effect. Because all you do is you form a set of equations. Oh, I say all you do, that's actually hard to do. But if you can form a set of equations which encodes the molecular level understanding, then you can simulate those equations, or you can show how those equations would play out at a tissue level. And you can basically use simulation, mathematical modeling and simulation, to bridge that gap. So it seems simple. I mean, it seems, for an engineer, I think, that's something, or a mathematician, or someone, physics, someone who works with mathematical models, that kind of thing is obvious. But it's not actually something that's used a lot in biology. And this growing area of systems biology, this is kind of one of the main research areas within systems biology, is bridging gaps between understanding and where the data exists. Okay, so I've got a few points here about why we'd want to model biological systems. And we've done two different efforts, and I'll just show you some results of the two different efforts. So when you think about mathematical models, you have all of the kind of limitations and sort of the pros and cons of each type of model. You know, in engineering, we're often seeking the simplest possible model which will capture which is rich enough to capture the behavior that we want to capture. The reason we want the simplest possible model is because if we could use mathematical analysis to understand that model, to basically manipulate that model and predict what that model will say about the system, then that's inherently more powerful than numerical simulation. But more complex models, sometimes we have to use more complex models that are not amenable to mathematical analysis. So we can't sort of use a pencil and paper and study it's too difficult to study the steady state behavior of those equations or the stability properties of those equations, and we have to resort to numerical simulation. So there's it's kind of this, always this, and we see this in engineered systems as well, this conflict between getting the simplest possible model which could admit analysis and you know, a model that's rich enough to actually represent what we want to represent. So we've done two different things, and actually we've done a lot of work in simplified models or hybrid models. So hybrid means it's a combination of two different types of dynamics, and our two different types of dynamics are discrete state and continuous state dynamics. So one particularly simple model to analyze is a switched linear system. Linear systems are amazingly easy to analyze, but rarely do biological systems fall under, rarely are they well modeled by linear systems because they're too complex. So switched linear systems is another type of model that we've used. And so we've done some work in basically building models which will bridge that gap between the particular system and the particular understanding and the system level effects. And then the second is really models which can be numerically analyzed. And typically the kinds of, well, the work that I've done here are mostly partial differential equation models, reaction diffusion models that model the kind of processes that we're talking about. And there, it's very rare that you can do like mathematical analysis on these models. Usually you resort to numerical simulation, or for identifying parameters, you generally have to do that numerically. So in the interest of time, I'm going to skip ahead. I had a little discussion of one of the systems we've worked on, which is a frog embryo. But I think what I'll do is jump over that and go back to the fly. So we'll just go down to the fly results here. And I'll show you the work that, I'll say a little bit about the model, and then I'll just show you the results in the last few minutes. So we've built a model for this system. So we had, so we go back to the fly. We had 4 proteins. And we basically decided to build a model with 10 signals, 10 state variables. 4 of them are the original proteins, 2 cytoplasmic and 2 membrane proteins. And then we introduced 6 what we call complexes. Now, proteins bind together. That's basically they react together and they can bind. Well, not much is known about these proteins in the fly. In fact, it's not even known whether or not they bind together. And so we're hypothesizing through these complexes that these proteins are actually binding together. And then we're going to test that hypothesis. If they don't bind, so we think about these complexes, like this one up here, disheveled, frizzled. It looks like a binding between those 2 proteins, but there may be some other protein involved that binds to each of them, so they don't bind together. But we're basically just using that complex as a mathematical entity which is representing a positive influence between those 2 proteins. So we're modeling positive influences, those arrows on Jeff's diagram, with binding. And it may not be binding, but if it represents what we want it to represent, it's okay with us. And then we form reaction equations. So basically, proteins and their complexes can join together, and they can join together with some forward reaction. They can break apart with a reverse reaction. And if we do this for all of the proteins and protein complexes, we end up with 10 reaction equations. And then we also propose there has to be some kind of movement of molecules, because that's sort of the data that we saw, that proteins localize asymmetrically from a homogeneous distribution. So what we did was we used kind of the least restrictive assumption. We don't know how the proteins are moving, so we proposed that the only way they're moving is through diffusion. We know that they do move through diffusion. There may be active motion going on, but our sort of justification in using diffusion only is that if we can make a model that works, that represents everything, only invoking diffusion as our motion mechanism, then it will only become easier later on if we get more data about molecular motors moving proteins through cells. It will only become easier to adapt the model to add convection terms, for example, to account for active motion. These equations, so this is just to show a little bit more about the equation, but basically we end up with a set of 10 fairly standard reaction diffusion equations. And the hard part, and the part that took the most time, is we hypothesized a structure. By a structure, we mean parameterized reaction diffusion equations. And then the hard sort of system-level mathematical problem is to identify a set of parameters: reaction rates, diffusion rates, reverse reaction rates. We had other interaction parameters, initial concentrations of the initial four proteins. So we had a system of 10. We have a system of 10 PDEs for each cell, partial differential equations for each cell, and then overall we have 37 parameters. So it's a fairly large problem. The PDEs are nonlinear, so a fairly large computational problem. So most of our effort went into identifying designing a good procedure for parameter identification. And we actually used a method from optimal control, which we have a particular, so this represents our system of equations. It's just represented in sort of state form. And then we form a cost function which penalizes difference between what the simulation does and what the observed data is. So that's the cost that we want to minimize, subject to the dynamics of the system. And this becomes a fairly standard, although large dimensional optimal control problem. So we solved it, and we came up with a set of parameters. And let me conclude by showing you the results. And I want to show you the results because I think they're neat, and they indicate really what you can do with mathematical modeling. Okay, so simulation exists on the left-hand side. So each of these represents a single cell in an infinite array of cells. The warm colors indicate high concentrations. The cool colors indicate low concentrations. So frizzled and disheveled, localized to the distal side, prickle and Van Gogh localized to the proximal side, which is not surprising because we actually included these as feature constraints in our cost function. And if you try the clone, so there's the experimental picture to remind you of what happens if you knock out frizzled. Knocking out frizzled in simulation is easy. We just remove all frizzled and frizzled containing complexes from a patch of cells, and we see the disruption of polarity as we'd expect. Removing Van Gogh, so X's represent the cells that don't have Van Gogh, and you see again the disruption of polarity on the left hand, on the proximal side. So we're able to reproduce, and we're able to reproduce the other data too. There's overexpression data, other mutant data. So we can go on to sort of these non-intuitive results, and this is where it gets interesting. So this was the third source of controversy that I told you about when we were introducing this. If you overexpress prickle, one might expect that you'd see less disheveled at the boundaries of the cells in which you've overexpressed prickle. So we simulated that. All of the cells with X's in have more prickle. And what you actually see, this is a plot where the higher intensity indicates more disheveled protein. You actually see the opposite of what you'd expect in simulation. And this is what matches the data. So this is an interesting result. It's kind of a non-intuitive result that is explained by the fact that that prickle-disheveled interaction, one can't just view that in isolation. It's actually a smaller subsystem that's part of a larger feedback loop. And the rest of the feedback loop here is overcompensating for the overproduction of prickle. So it's it indicates that the system is more complex, and it's a very non-intuitive result. Other results, which, so we were actually able to go back and try to, thanks, that's a lot better. So we went back and we tried to reproduce some of these other sources of controversy. And from that, we were actually able to go back to the biology and predict bindings that we didn't know about before. In particular, through our hypothesis, and I won't explain the details, but what we did was we predicted through the mutant behavior of this sort of second frizzled allele that I talked about earlier, that frizzled and disheveled are actually binding with each other. And we used the model to predict what the system would look like if we did the experiment. And then we went back and did the experiments. We, meaning one of Jeff's students, went back and replicated in the lab what we'd done on the computer. And he was able to match it perfectly. And I think that's why, actually that's honestly why this paper got published in a biology journal, because you can't just publish a model unless it gives you some new results. And this was a new result that we concluded, which was a direct consequence of this modeling that we did. Finally, let me just go to sort of what's interesting. Well, actually, I have one more thing to say before I go on to the last point, which goes back to this kind of controversial community. So Peter Lawrence is a developmental biologist at Cambridge. He's very well known. He's a superb biologist. He works in planar cell polarity. And we'd sent him a draft of our paper to review before it had, or a manuscript before it had been published. And he didn't believe the model. In fact, he had published a competing model earlier, which was one of these factor X, factor Z things. So he said to us, all right, I've done a set of experiments. I'm going to send you the setup of the experiments, but I'm not going to send you my results. You simulate my experiments on your model and send your simulations back to me, and I'll tell you how good your model is. So, and these were really weird experiments. So they were overexpressing frizzled in a background of cells that didn't have any Van Gogh, or a clone of cells that had no frizzled and no prickle, etc. And so we simulated all three. Those are the results. We sent the results back to him, and we matched all three. So this is just one of the experiments. He does work on the fly abdomen, so the images look a bit different. But basically, we were looking for matching the polarity of cells around the clone, basically whether or not we got this, what we called non-autonomous phenotype. And so that was actually really nice, because we've, when you want to do model identification, you want to make sure that you've sort of separated your data into data that you use for the identification process and data that you test on. And so we did that, but here was a really independent set of experiments that allowed us to validate the model. Okay, so finally, So insights into autonomy, et cetera. So we got some insights. We've shown, I think, that this model is plausible. We haven't shown that it's the correct model, because we may get data tomorrow which disproves the model. I don't think we will, but we might. But this is the way it goes in systems biology, is that you're always updating models as you get new hypotheses and as you get new data about the system. Okay, so finally let me conclude on this global directional cue. I mentioned this at the beginning of the talk. So this protein fat, if we have a model, we have to, it has to explain the difference between these two clones. And remember, the difference is that in some clones of fat, polarity is propagated nicely. In some, global polarity is disrupted, but local polarity is maintained. So what we proposed is that in the absence of this global directional cue, cells are getting, the information that they're getting to determine their polarity is only coming from their neighboring cells. And how they interact with their neighboring cells is determined by their geometry. You know, the number, if we saw regular arrays where cells are basically hexagonal, they have 6 neighbors. But if their geometry is disrupted, then they may have 5 or 4 neighbors, and you can kind of actually, well, I don't know if you can see it from there, but you can see the faint outlines of the cells. And it's actually, in that clone up there, we have fairly regular hexagonal geometry for each cell. But in this clone here, actually the point, the sort of start of this vortex, is a long, thin cell with disrupted geometry. So by looking at these images, we proposed that cell geometry is a deciding factor in polarity in the absence of the global directional cue. So to test that, we had to go back and now build irregular geometries. So we looked at images. We formed irregular grids from those images. We simulated our model on those irregular grids now. So that just shows a wild-type polarity, which we'd expect. We looked at, so there's the hair polarity. We looked at clones. So this was a frizzled clone. We wanted to replicate the same behavior. So we got up to the same level with irregular geometries as we'd done before with regular. And then we started simulating the fat clones. And so here's the match. And it's a pretty good match. So here you see the disruption. It's not really a swirly pattern. It's more like a starburst pattern. That means that we probably still have a little more optimization to do in our parameter set. But we're matching the disruption in polarity and the non-disruption in polarity in these fat clones really nicely now. So that, again, it doesn't prove our hypothesis is correct, but it validates our hypothesis that geometry is a deciding factor in polarity. Okay, so these have all been sort of very intricate little results, right? You're sort of wondering, well, why would you care about fly wings? And we talked about that at the beginning, that really this community is trying to work on smaller problems to really understand larger problems. And so, so in actually sort of these successful modeling efforts, it shows the role, I think, that mathematics and modeling and control and engineering can play in understanding biological systems. So we've done that and used that to predict a few things. And I think sort of going forward, the last 2 points are the most interesting, is that the success of this model now has brought in other possibilities for us. So we're now working on a lymphoma project. This is really exciting. So we have, again, it starts with a hypothesis of different oncogenes that are, so cancer-causing genes or cancer-related genes that are interacting with each other that may cause lymphoma, which is a particularly deadly form of cancer. And it's neat because we have both mouse data for this as well as human patient data from the Stanford School of Medicine is connected with the Stanford Hospital. And so that's a project that we're working on now, same type of modeling effort. And then we've just started a new project with Lawrence Berkeley Labs. There's a really wonderful collaboration between LBL and a number of the biologists and actually some of the engineers here on campus, which is doing like a full-scale concerted effort to try to understand the early Drosophila development. So this is earlier than I was looking at with flies. It's really when the embryo is starting to form and when it starts to differentiate between, from stem cells to different kinds of cells that form different parts of the fly body. So thank you very much for your attention and questions.
Two questions, please. Yes, let me give you the microphone because we're being recorded, as you know, or rather video.
So obviously it's wonderful work. Let me play devil's advocate for a minute here. And the question that I had is that it looks to me like if you vary the parameters enough, you can get any kind of pattern that you've seen before.
Right.
So I was kind of wondering how much data you have to train your algorithms to reproduce what you know already is the case before you can make a prediction.
Yeah, now that's a very good question. And it's not only how much data, but what types of data. So we needed data that basically allowed us to reproduce all of the independent degrees of freedom of this system. But we don't have a nice, it's not a linear system. We can't look at eigenspaces and stuff. So we had to do it manually. So what we did was we started with the smallest amount of data that we thought was important to differentiate two different behaviors. And then we gradually started building up. And we had Jeff sitting there at our side saying, who knows the system very well. So it's still a very manual process for understanding how much data and what types of data you need in your identification. Now we know for this system, but there's not, and I think this is very important in systems biology, is developing principled ways of doing this for large-scale, complex, nonlinear systems. And to say, yeah, another point about that, I guess, it really did start from like using the least amount of data possible and then just building up from that, realizing that we're not going to reproduce a certain phenotype with the data that we had, so we have to include that in the identification process. A point about varying parameters, though, you mentioned that at the beginning of your question, and I didn't mention this and I should, is that we don't only want a parameter set that works. So, if we think about the cost function over the parameter space, we get these kind of low peaks, so a very low cost. But if there are sharp walls, so you vary the parameters slightly and you lose the phenotype, that parameter set is no good. So our identification process, this optimal control method, had to include in it something that encoded the robustness of the parameter set. So you want to sort of search low valleys are much better in this parameter, in this cost function, than our sharp peaks. So you want to be able to vary the parameter sets and still reproduce the same behavior. And that's a requirement of the process.
Interesting point. Another question from the floor? Yes. Hi. Hi, Brent.
Nice to see you. My question is sort of along the same lines, and I guess the question's very simple. I understand that the work behind it might be monstrous, and apparently there are other models, like your challenger Lawrence, as you said, had another model, right, for predicting also that behavior. Right. Did you try any of these other models and find that falsify them, or perhaps
well, his model, so that's a good point. So Lawrence's model was a biological model, so along the lines of the influence diagram. We didn't simulate it, so we didn't form a set of equations which simulated his model. And that's mainly because it invoked additional factors which we didn't have biological evidence for. So a factor X and a factor Z. And our goal was to show in this first part of the project that the simpler model, which didn't invoke these additional factors, it was sufficient to be able to explain the data. Now, since then, there's been another paper published which has it's a similar model to ours, but it has another protein involved that we know is involved. We just didn't include it because we didn't have, like, if you remove it, you don't disrupt the overall polarity. And now, so with that data and that model, we've simulated that model, and we've been working together with those guys. So yeah, it really is. I think, despite what I said, it is a very collaborative effort among people in the community. Yeah.
Just hang on. Sorry, I'm sorry to slow you down with the microphone.
Just one thought there is, and I love what you showed. I think it's really wonderful. But one thought there is, if you play devil's advocate, is that you can take any model. If you just tweak the parameters in the right directions, you can, get to the same pattern with a completely different model. And I'm just wondering how robust those models are. And if you really can do what was suggested, to take a different model that we would think is wrong, and there's no way you can get to the same result.
So you can't take any model. So that's pretty clear. You have to have a certain, you have to start with a class of models within a certain structure. And so our goal was to try to find the simplest possible model. So anything that includes more proteins and the diffusible factors where there's not even any evidence that proteins are diffusing from cell to cell. I mean, in our model, we don't even need that. They're only diffusing within their own cell, and they're interacting with neighboring cells. So within a certain class of models, we were looking for the simplest possible model. And it's true. Other models within that class, you could find a set of parameters which would work. But then, so we weighed it up by looking for the simplest possible model, which would explain all of the data. And also we could use in predictive. If we used it for predictions, we got the right prediction when we went back and experimentally verified that.
Yes, please ask a question.
So you mentioned that you use, small organisms like Drosophila and stuff because of the short generation times. Have you ever considered doing these types of modeling on mammalian cell cultures in vitro, which also have short generation times?
Yes. I'm not a biologist, although I've been learning a lot about it. But now Jeff's lab is doing mice. So doing mice meaning they're looking actually at that cochlea problem. So they're looking at the formation of cells in the inner ear of mice, and they're both doing in vivo and in vitro experiments. So the in vivo ones take longer, but in vitro they can do experiments with a shorter. That's a good point. Yeah.
Good question. Well, maybe especially the folks that really understand this in great depth could come forward and chat with Claire after we give her a warm round of applause. Thank you very much.